Okay, today we're going to start our unit on genetics, and we're going to learn about Gregor Mendel and his importance to genetics. So here are some terms. Trait, any characteristic that can be passed on from parent to offspring. Trait, characteristic, you can use those words interchangeably. An example of one of your traits would be maybe that you have blue eyes or brown eyes or green eyes or brown hair or freckles. Heredity is the actual passing of traits from parent to offspring, and genetic is the study of heredity. Students tend to confuse the word heredity and the word genetic. Genetic is the study of heredity. Like I mentioned, Gregor Mendel is the father of genetics. He was a teacher. He did all of his research with pea plants because they were easy to study and because their traits were known as being pure. He was the first one to come up with some basic laws of heredity. Again, he studied the purebred traits in peas. He knew that they were purebred, which means that if you had a tall plant, the only trait that plant could pass to its child is being tall. It could never pass being short because it's pure. The only gene it has is for tallness when it comes to height. He learned that there were dominant genes versus recessive genes. The dominant one, just like in a dominant personality, it's going to be expressed. It's the one that gets to show up. The recessive gene is weaker. It can't show up. It can't overpower that dominant gene. He presented his information. They pretty much ignored it. It wasn't until after he had passed away that his work was rediscovered, and he's now known as the father of genetics. Again, he mentioned he worked with the peas because they were easy to work with. He had pure traits, and he looked at 7 or 8 different traits. It depends on which book you look at, and we're going to look at those in a minute. So they were pure, so they were purebred for all of those traits that he looked at. And he crossed the pure dominant with the pure recessive, and he did this thousands and thousands of times and kept a very good records. One thing you have to remember, when Gregor Mendel was doing his work, they knew nothing of genes nor DNA at the time. So that's what makes his work kind of exceptional. Just a little bit about peas. You know that pollen fertilizes the egg in a pea. We talked about that. Pea plants, or at least most plants, actually reproduce sexually, which means pollen, aka sperm, gets to an egg. There is distinct gametes or sex cells. Again, you have the pollen and you have the egg. Pea plants normally reproduce by self-pollination as opposed to cross-pollination, so pea plants can pollinate themselves. Fertilization is the uniting of the male and female gamete, and a cross is combining gametes from parents with different traits. So what Mendel did, he did a lot of crossing. He crossed a tall plant with a short plant, a yellow plant with a green plant. So crossing, it's kind of meaning like sex. He had a tall plant have sex with a short plant. In plants, the male part is known as the stamen, and it's made up of the anther and the filament, and the female part is known as the pistil, and it has the stigma, style, and ovary. The stigma is sticky so that the pollen can stick to it. The pollen then would travel down the style and get to the ovary, and then fertilize the egg that's there and make a baby plant or seed. And again, here's a definition for self-pollination versus cross-pollination. When it comes to plants, some plants will have a male flower and a separate female flower on the same plant. Some plants, the male and female part are together on the same plant, just like in this diagram. Then there are plants that have the whole plant is either male or the whole plant is either female. So plants can actually be kind of complicated to study. When it came to Mendel's peas, he had to do a controlled experiment. He couldn't just have pollen flying everywhere and just guessing what pollen landed on the stigma to fertilize the egg. So to have a controlled experiment, he cut off the male parts. He then took a tiny paintbrush, put pollen, got the pollen off the male part, and brushed it onto the stigma or the female part. So with Mendel's experiment, he did the pollination. He did the self-pollination himself. He could not have pollen flying around everywhere. And some of his results, you can see here, his first generation he called the P generation, meaning the parent generation or parental generation. Again, he had a pure tall, crossed. We literally mean, we write an X. In other words, the tall plant had sex with the short plant, and it's a pure tall plant and a pure short plant. They had the babies, The baby generation is known as the F1 generation, and every single one of them were tall, if we're talking height. They were always tall. He never could get a short plant in that generation. And that's what led him to say that there were dominant genes versus recessive genes. And that short must have been recessive and it just could not be expressed. It didn't disappear, it just could not be expressed. And this kind of surprised Mendel. He thought he was going to get medium-sized plants in his F1 generation. Well, then he got this idea, what if I take my F1 generation and cross them among themselves? Let's see what happens. And that's where he came up with his F2 generation. And this is where we had that recessive allele come back. You still had the tall allele, but the recessive allele was able to be expressed this time, and we'll see why later. So when he went from when he had the P1 or P cross, there was a trait that disappeared. The recessive trait disappeared. When he did his F1 cross and looked at those children, he noticed that that recessive trait came back. Here's just a list of some of those traits he looked at. And here's a list also. And I want to look at this chart. And we're going to write out some crosses. In genetics, letters are used to represent genes. We can't really see the genes, but we use a letter to represent them. And we use a capital letter for dominant and a small letter for recessive. So he looked at seed shape, seed color, pod shape, pod color, seed coat color, flower position, plant height, and flower color. So let's just say for the first trait there, seed shape. What Mendel did, and he had pure plants. So he had a pure round, and it was crossed with a pure recessive. Okay, the pure round was crossed with the pure recessive. Round is dominant. So we have two capital R's. Wrinkled's recessive, so we use the two little r's. Those big R's literally overpower the little ones. We have to use two letters because everybody in here, and all plants, you have two parents. So the first R, that was a gene from one parent. This was a gene from the second parent of this flower. And then you have the next parent. Maybe this is the father. The father had to have one gene from mom and one gene from dad. Well, then you cross these two plants, and the only gene that will be expressed is going to be the dominant one. You get one from each. So the baby that's being made in this case, because we had big R crossed with little r, or pure round crossed with pure wrinkled, in this case, I'm doing seed shape. So the first parent can only give a big R. The second parent can only give a little r. So you have a child that has big R, little r genes. And the way that this plant's going to look is round. We could do that with all 8 of these traits. So seed color, big Y, crossed with little y, or pure yellow crossed with pure green. The child would be big Y, little y, or yellow. With Mendel's experiments, he had, as you notice, 3 different generations. He experimentally crossed different strains to develop what were known as hybrids. A hybrid is a child that is a little different than its parents. So what he started with, he called his P1 generation, which was known as his parental generation. Pure tall, crossed with pure short. That's what I just did on the last slide, but I did it with seed shape. You had this cross where we used the letters to represent genes. So we had 2 capital T's crossed with 2 little t's to get a big T, little t. This child, in this case, would be tall. Again, you could write it out in words. Pure dominant crossed with pure recessive of the same trait. Then, like I mentioned earlier, he decided to cross the hybrids and analyze the result. So he wanted to take these children made here and cross them among themselves. He called this generation the F1 generation, and F stands for filial, which means son in Latin. So he took the first set of children, F1, and crossed them among themselves. And as you can see, they have a big letter, little letter. They're not pure anymore. There's no pureness. He called this heterozygous. So he took a heterozygous tall, crossed with a heterozygous tall, or if you were to write it out in its genes, big T, little t, crossed with big T, little t. And a little bit later, we're going to find out what kind of results he would have come up with when he did this. And he did this with every single trait, all 7 to 8 of those traits. Here are some more vocabulary words. A few of these we've talked about already. A trait is a specific characteristic. A hybrid is an offspring that is a little bit genetically different than its parents. Genes are chemical factors that determine traits. They're made from your DNA. And then allele, different forms of a gene. The tall form, short form, the round form, the wrinkled form. And you're going to find out with humans it's just not quite that simple. But with these pea plants, it was that simple, and that's why he used pea plants. Every single trait only had 2 alleles. Each organism has 2 alleles for every trait. I mentioned that earlier. That is because you get one from mom and you get one from dad. With humans, we have a few traits that only have 2 alleles, and we're going to talk about those at a later time also. And remember, those genes are on your chromosomes, and your DNA came together to make your genes. Like it mentions here, though, there are genes out there that have 3 alleles. I already mentioned your hair color, your skin color, your eye color. There's just more. There's not just this or this. Eye color, you have blue eyes, you have brown eyes, you have green eyes. Hair color, you have black hair, you have brown hair, you have blonde hair, you have red hair, and everything in between. Okay, so again, we're going to do just a quick review. His P1 generation, they were pure, so he did what was known as a P1 cross. So this would be the P1 cross. Pure tall, crossed with pure short. No matter what you get, the only thing that parent can give to his child is a big R. The only thing that parent can give to its child is a little r. We're using the trait of seed shape, round versus wrinkled. This child would be round. Then for his F1 generation, his first set of children, he crossed with themselves. He did what was known as a F1 cross. Big R, little r, crossed with big R, little r. So he took what was called a heterozygous round, crossed with a heterozygous round. They're not pure anymore. They're not pure. So that's known as heterozygous. And then here's just another quick review. Strong hereditary traits cover the weak ones, so the dominant covers the recessive. Dominant traits are represented by the capital letters. Recessive traits are represented by lowercase letters. Rule of dominance. The trait that is observed in the offspring is the dominant trait. The trait that disappears is the recessive trait. And in his P1 cross, to make his F1 children, the recessive parents' traits disappeared. Genotype, phenotype. I haven't used these terms yet. Phenotype is the way an organism looks. It's physically the way you look. Phenotype, physically the way you look. Red hair, brown hair. The pea plant that was tall. The pea plant that had round seeds. The pea plant that had wrinkled seeds. Genotype are those genes that we use to be represented by letters. Big A, big A. Big A, little a, little a, little a are genotypes. Heterozygous versus homozygous. Heterozygous is when those 2 letters are different. They're not pure. Homozygous is a term that stands for pure plants. Big A, or little a, little a. And it does not matter what